Imagine holding a secret in your hands, a secret so strange, so utterly impossible, that the greatest minds of an entire era refused to believe it was real. Welcome to Bedtime Biology, where science meets serenity. Each night, we explore the quiet wonders of the natural world, calming your mind and sparking your curiosity. Tonight, we're diving into the cool, dark waters of Eastern Australia to meet a creature that shouldn't exist, a mammal that lays eggs, a hunter that sees with electricity, an animal with venom, a duck's bill, a beaver's tail, and fur that glows in the dark. This is the story of the platypus, and every fact you're about to hear is absolutely true. The year is 1799. In the hallowed halls of the British Museum, a collection of the world's finest naturalists gather around a wooden crate that has just arrived from the distant colony of New South Wales. Inside, packed carefully in preservatives, lies a creature unlike anything they've ever seen. At first glance, it appears to be some kind of elaborate hoax. Surely, someone has taken the bill of a duck and sewn it onto the body of a mole or beaver. The stitching must be here somewhere. They examine it closely, searching for thread, for glue, for any sign of deception. But there is none. The bill is attached by flesh and bone. The webbed feet grow naturally from limbs covered in dense, chocolate-brown fur. The flat, paddle-like tail seems to belong to an entirely different animal, yet it too is genuine. And strangest of all, beneath the tail, where one might expect to find the typical mammalian arrangement of reproductive and excretory openings, there is only a single hole, more reminiscent of a bird or reptile than any known mammal. George Shaw, the keeper of the Natural History Department, would later write in his official description that he had examined the specimen with great care, fully expecting to discover evidence of fraud. The creature was simply too bizarre to be real. It combined features from so many different animals that it seemed more like a mythological chimera than a product of nature. But it was real, perfectly, impossibly real. The specimen had been collected along the rivers near Sydney, where colonists reported that these strange animals lived in burrows along the banks and spent their time diving in the water, searching for food. The local Aboriginal people, of course, had known about these creatures for thousands of generations. They called them by many names across different language groups: Boondabura, Tambrit, Malingong, and many others. To them, the animal was not strange at all, but simply another thread in the rich tapestry of life that filled their continent. But to European science, the platypus represented something unprecedented. It would take decades of study, countless specimens, and the development of entirely new branches of biology before scientists could begin to understand what they were looking at. Even today, more than 2 centuries after that first preserved body arrived in London, the platypus continues to surprise us, revealing new secrets with each generation of researchers who dedicate their lives to studying it. The platypus, known scientifically as Ornithorhynchus anatinus, which roughly translates to duck-like bird snout, is not just an oddity. It's a window into deep evolutionary time, a living link to the ancient past when the great tree of mammalian life was first branching in different directions. To understand the platypus is to understand something fundamental about how life adapts, survives, and finds its own unique path through the endless challenges of existence. Let's begin with the most obvious question. What exactly is a platypus? Picture a mammal about the size of a house cat. Males typically measure between 15 and 24 inches from the tip of their bill to the tip of their tail, weighing anywhere from 3 to 6 pounds. Females are slightly smaller, measuring 14 to 21 inches and weighing between 2 and 4 pounds. These dimensions can vary considerably depending on where a platypus lives. In the colder southern regions of Australia and Tasmania, platypuses tend to be larger, while those living in the warmer northern areas are generally smaller, a pattern known as Bergmann's rule. The first thing anyone notices about a platypus is, of course, its bill. But this is no hard, brittle structure like the beak of a bird. Instead, the platypus bill is soft and rubbery, flexible to the touch, with a texture that has been compared to fine suede leather. It's dark in colour, almost black, creating a striking contrast against the rich brown fur of the body. The bill itself is quite large in proportion to the animal's head, broad and flattened, giving the platypus an almost comical appearance when viewed from above. But this bill is far from a joke. It represents one of the most sophisticated sensory organs in the entire animal kingdom, though we'll explore those remarkable capabilities in greater detail soon. For now, simply note that the bill contains no teeth. Adult platypuses are completely toothless, though they're born with vestigial teeth that disappear early in life. Instead, they have hardened pads of keratin, the same material that makes up our fingernails, which they use to grind up the food they collect. The platypus body is streamlined and torpedo-shaped, perfectly designed for moving through water. Every aspect of its anatomy serves this aquatic lifestyle. The fur is perhaps the densest of any mammal on Earth, with two distinct layers. The outer layer consists of long guard hairs that protect the soft underfur beneath, which remains dry even after hours of submersion. This remarkable waterproofing is achieved through the structure of the fur itself and the natural oils that coat each hair. Between the fur and the skin, the platypus traps a thin layer of air that provides both insulation and buoyancy, allowing it to stay warm and maintain the right depth in water that can be near freezing. The coloration of a platypus varies from dark brown to reddish brown on the back and sides, with a lighter silver-grey belly. Under each eye, there's a distinctive patch of white fur, giving the appearance that the eyes are larger than they actually are. When a platypus dives underwater, these white patches may create a confusing visual signal for predators or prey, making it harder to discern the animal's true size or direction of movement. The feet of a platypus tell the story of an animal caught between two worlds. The front feet are the primary means of propulsion in water. They're broadly webbed, with the webbing extending well beyond the claws. When swimming, the platypus uses these front feet in an alternating paddle motion, very much like a swimmer doing the breaststroke. The hind feet are only partially webbed and serve primarily as rudders, along with the broad, flat tail, which also acts as a stabilizer during swimming and a fat storage organ during times of plenty. But here's where it gets interesting. On land, that extensive webbing on the front feet would be a hindrance, making it difficult to walk or dig. So the platypus has evolved a remarkable solution. The webbing can retract. When a platypus emerges onto the bank, the webbing folds back, exposing strong claws that are perfect for digging burrows and moving across uneven terrain. The animal walks somewhat awkwardly on land, moving with a distinctive reptilian gait because its legs extend out from the sides of its body rather than directly underneath. another ancient characteristic it shares with its distant evolutionary relatives. The tail of a platypus is broad, flat, and covered with fur, quite unlike the naked, scaly tail of a beaver, despite the superficial similarity in shape. This tail serves multiple purposes. In water, it acts as a rudder and stabilizer. On land, it helps the animal maintain balance. And year-round, it serves as a storage depot for fat reserves, becoming quite plump when food is abundant and slimming down during lean times, or when a female is raising young and unable to forage as much. One of the most peculiar anatomical features of the platypus, and one that contributed greatly to early confusion about its classification, is the cloaca. This is a single opening that serves for reproduction, urination, and defecation, much like what we see in birds and reptiles. The very word monotreme, which refers to the group that includes platypuses and echidnas, means single opening. This is a primitive trait, one that the earliest mammals possessed before evolving separate openings for different functions. The platypus, in retaining this feature, gives us a glimpse of what our own distant ancestors might have been like, hundreds of millions of years ago. Inside, the platypus body reveals even more curiosities. The animal maintains a relatively low body temperature for a mammal, averaging around 32 degrees Celsius, compared to the 37 degrees typical of most placental mammals. Yet despite this lower baseline, platypuses can maintain stable body temperatures, even when diving repeatedly into water as cold as 4 degrees Celsius. This is accomplished through that remarkable fur, behavioral adaptations like limiting dive times, and metabolic adjustments that increase heat production when needed. The skeleton of a platypus shows a fascinating mixture of mammalian and reptilian features. The shoulder girdle contains extra bones not found in other living mammals, a feature it shares with its extinct ancestors and with modern reptiles. The ribs, too, have some unusual characteristics, including rudimentary ribs on the neck vertebrae. These skeletal peculiarities aren't just interesting trivia, they're important clues to understanding the deep evolutionary history of all mammals. When you look at a platypus, you're seeing an animal that has retained features from various points in mammalian evolutionary history, while also developing highly specialized adaptations for its unique lifestyle. It's a mosaic, a patchwork, a living museum of traits that in most mammals have been lost or transformed beyond recognition. But the truly extraordinary features of the platypus go far beyond what we can see on the surface. To really understand this animal, we need to dive beneath those dark Australian waters and experience the world as a platypus does, through senses we humans don't possess and can barely imagine. Close your eyes for a moment and imagine you're swimming in complete darkness. Not just the darkness of night, but the absolute blackness of deep water clouded with sediment. Your eyes are sealed shut, your ears are covered, even your nostrils are clamped tight. You're gliding along the muddy bottom of a river, your body moving with practiced ease through the cold current. How do you find food? How do you navigate? How do you avoid obstacles and predators? If you're a platypus, you use electricity. The discovery of electroreception in the platypus is a relatively recent chapter in the animal's scientific story, and it represents one of the most remarkable examples of sensory adaptation in the mammalian world. For most of the platypus's known history to Western science, researchers assumed that the animal's bill was primarily a touch-sensitive organ, useful for probing in mud and detecting prey through mechanical contact. And while the bill certainly is sensitive to touch, that turns out to be only part of the story. The breakthrough came in the 1980s, when a team of German and Australian researchers conducted a series of elegant experiments. They observed that platypuses could locate and capture prey, even in complete darkness, even when the prey was motionless. They found that platypuses could detect and respond to weak electrical fields in the water. And when they examined the structure of the bill under high magnification, they discovered something extraordinary. Alongside the mechanoreceptors they'd expected to find, the bill contained tens of thousands of specialized electroreceptors. These electroreceptors are concentrated in the skin of the bill in distinctive stripe patterns, with approximately 40,000 of them distributed across both the upper and lower surfaces. They're most densely packed near the edges of the bill, where sensitivity needs to be highest. Each receptor is connected to a mucus-filled pore that opens to the water, allowing it to detect even the faintest electrical signals. But how does this actually work? Every living thing generates tiny electrical fields. When a muscle contracts, it produces a small electrical pulse. When neurons fire, they create electrical signals. Even the simple act of a shrimp flicking its tail creates measurable electrical activity in the surrounding water. These signals are incredibly weak, measured in microvolts, millionths of a volt. To put that in perspective, a platypus can detect electrical fields weaker than what a standard household AA battery produces, diluted across a vast volume of water. The platypus bill can detect these signals at a distance of up to 15 to 20 centimeters, and possibly even farther under ideal conditions. When a platypus is foraging, it swings its head from side to side in a characteristic scanning motion, sweeping the bill back and forth through the water. This movement serves several purposes. It increases the area being searched, much like how we might sweep a flashlight back and forth in the dark. But more importantly, it allows the platypus to compare the strength of electrical signals hitting different parts of the bill at different times. By detecting these subtle differences in signal strength and timing, the platypus can calculate both the direction and the distance to its prey with remarkable precision. It's a form of bioelectrical triangulation, allowing the animal to create a three-dimensional map of its surroundings based purely on the electrical signatures of living things around it. But electroreception is only part of the story. Remember those mechanoreceptors we mentioned? There are even more of those than there are electroreceptors. The bill contains approximately 60,000 mechanoreceptors, specialized cells called pushrod receptors that detect changes in water pressure and motion. These receptors are so sensitive they can detect the displacement of water caused by the tail flick of a small crustacean from a distance of up to 50 centimeters. Here's where it gets truly fascinating. The electroreceptors and mechanoreceptors don't work independently. They're integrated in the platypus brain in a way that creates a unified sensory experience. When a shrimp flicks its tail, it creates both an electrical pulse from the muscle contraction and a physical disturbance in the water from the movement. The electrical signal travels essentially instantaneously through the water, while the mechanical pressure wave travels at the speed of sound in water. By comparing the tiny time difference between when the electrical signal arrives and when the mechanical signal arrives, the platypus can calculate the distance to its prey with extraordinary accuracy. Think about that for a moment. the platypus brain is performing complex calculations based on the integration of two entirely different types of sensory information, arriving at slightly different times, to create a precise three-dimensional understanding of where prey is located in the murky water around it. And it's doing this continuously, in real time, while swimming, while holding its breath in complete darkness. The neural processing required for this is substantial. About 2/3 of the platypus's somatosensory cortex, the part of the brain responsible for processing touch and other body sensations, is dedicated to processing information from the bill. In the human brain, a similar disproportionate amount of neural real estate is dedicated to our hands, which are our primary organs of touch and manipulation. In the platypus, the bill dominates the sensory world. Interestingly, this remarkable sensory system may have evolved in connection with another unusual feature of the platypus, the loss of teeth. The platypus's closest living relative, the echidna, also has electroreceptors, though far fewer of them. But ancient platypus relatives, species like Obdurodon that lived millions of years ago, had functional teeth and may have hunted differently, possibly taking larger prey in open water, where vision could still play a significant role. As the lineage leading to modern platypuses specialized for bottom feeding in murky water, teeth became less useful, and the electroreceptive sense became more developed, a trade-off that ultimately produced the unique creature we see today. The discovery of electroreception in the platypus opened up an entirely new area of research and raised countless questions. How is the information from all those thousands of receptors processed? How does the platypus brain distinguish between the electrical signature of a shrimp versus a worm versus a caddisfly larva? Can platypuses detect the electrical fields generated by other platypuses? And if so, might this play a role in communication or reproduction? Some of these questions are beginning to be answered. Research has shown that neurons in the platypus's sensory cortex are arranged in alternating stripes, with electroreceptive neurons adjacent to mechanoreceptive neurons. At the borders between these stripes, there are neurons that appear to integrate both types of information, creating a unified sensory map. It's elegant, efficient, and unlike anything found in other mammals. But here's a sobering thought. Platypuses famously do poorly in captivity, and one of the reasons appears to be related to this exquisitely sensitive electrical sense. The pumps and filters used to keep aquarium water clean generate electrical fields that can overwhelm and distress a platypus's sensory system. It's like being forced to live in a room with a fire alarm constantly blaring. Some platypuses in early captive breeding programs died from the stress of electrical pollution in their tanks before researchers understood the problem and developed better filtration systems with electrical shielding. This vulnerability highlights an important truth about specialized adaptations. The same features that make a species supremely suited to its natural environment can become liabilities when that environment changes. As we humans continue to alter the rivers and streams of eastern Australia, filling them with pollutants, sediments, and artificial electrical fields from various infrastructure, we may be making these waterways less hospitable to platypuses in ways we're only beginning to understand. Imagine, if you will, that you're a 19th century naturalist exploring the rivers of New South Wales. You've been fortunate enough to capture a live platypus, a rare achievement. You're examining this extraordinary creature, noting every detail of its anatomy, when suddenly there's a sharp pain in your hand. The platypus has struck you with its hind leg, and on the heel of that leg is a sharp, hollow spur that has penetrated your skin. Within minutes, the pain intensifies. It's not like any pain you've experienced before. It's described by those who've suffered it as excruciating, worse than broken bones, worse than bullet wounds, worse than almost any pain short of the most severe injuries. The pain spreads up your arm and your hand begins to swell. No amount of morphine provides relief. The pain will persist for days, possibly weeks, and in some cases, it leaves a lingering sensitivity to pain that can last for months. Welcome to the world of platypus venom, one of the most painful substances produced by any mammal. Only male platypuses are venomous. Each male has a hollow keratinous spur on the inside of each hind ankle, connected by a duct to a crural gland in the upper leg. These glands, shaped like kidneys, produce venom that accumulates in the spur and can be injected when the platypus strikes. The spur itself is sharp and curved, resembling a large thorn, measuring roughly half an inch in length. It normally lies flat against the leg, but when needed, the platypus can rotate it perpendicular to the limb, allowing for a more effective strike. Female platypuses are born with rudimentary spur buds, but these don't develop and eventually fall off before the end of their first year. The factor that only males possess functional venom spurs and that the venom glands swell to their largest size during the breeding season tells us something important about the function of this venom. It's not primarily a defense against predators, though it certainly can serve that purpose. Instead, it appears to be a weapon used in combat with other males during the breeding season. Male platypuses are territorial and compete aggressively for access to females during mating season. When two males fight, they wrap their hind legs around each other, each attempting to drive his spurs into his opponent. A successful strike can temporarily paralyze a rival, leaving the victor free to mate, while the loser retreats to recover. Some researchers have found wild male platypuses with puncture wounds on their bodies during breeding season, evidence of these violent encounters. The venom itself is a complex cocktail of at least 19 different proteins and peptides. Many of these are unique to the platypus, though others show interesting similarities to toxins found in snakes, spiders, sea anemones, and other venomous creatures. This is an example of convergent evolution, where completely unrelated organisms independently evolve similar solutions to similar problems. In this case, the problem of how to incapacitate another animal. The major components of platypus venom fall into three categories. The first are defensin-like proteins, molecules that are normally part of the immune system, but that the platypus has co-opted and modified for use as venom. The second are C-type natriuretic peptides, which affect blood pressure and cause blood vessels to dilate. The third is a nerve growth factor, which paradoxically seems to be responsible for much of the pain. One particularly unusual feature of platypus venom is the presence of D-amino acids. Almost all of life on Earth uses L-amino acids to build proteins, with the molecules oriented in one particular direction. D-amino acids are mirror images of L-amino acids, and they're extremely rare in natural biological systems. Platypus venom is the only known example in mammals of proteins containing D-amino acids, a feature that makes the venom more resistant to breakdown by the victim's enzymes and may contribute to its long-lasting effects. The venom causes a cascade of effects in victims, it triggers a massive release of inflammatory compounds, causing severe swelling that can spread far beyond the site of the sting. It affects blood pressure, sometimes causing dangerous drops. It causes what's called hyperalgesia, an increased sensitivity to pain that can persist long after the initial injury. And perhaps most remarkably, it appears to work through pain pathways that are resistant to conventional painkillers. Even morphine, one of the most powerful pain medications available, provides little relief from platypus venom. There are fewer than 20 documented cases of platypus envenomation in medical literature, but several of these have been studied in detail. One particularly well-known case involved Keith Payne, a decorated Australian war veteran and recipient of the Victoria Cross. In 1991, Payne attempted to rescue a platypus that had become stranded and was struck by the animal's spur. He later described the pain as worse than being hit by shrapnel in combat. A month after the incident, his hand was still painful and difficult to use. 3 months later, he still experienced significant functional impairment. Another detailed medical case from 1992 described a man who was envenomated on the hand. Standard analgesics provided no relief. Morphine administered both intermittently and by continuous infusion was ineffective. The only treatment that worked was a regional nerve block, essentially numbing the entire arm by blocking the nerves that carry pain signals from the limb to the brain. Even with this treatment, the patient required narcotic pain medication for several days and spent 6 days in the hospital. The envenomated area remained painful and swollen with limited movement for 3 weeks, and significant functional impairment persisted for 3 months. There is no antivenom for platypus envenomation. Treatment consists of pain management and supportive care, waiting for the venom's effects to gradually subside as the body slowly breaks down and eliminates the toxic proteins. Interestingly, while platypus venom is excruciatingly painful to humans and can cause severe symptoms, it's not lethal to us. There are no recorded human deaths from platypus envenomation. However, laboratory studies on smaller animals tell a different story. When rabbits and mice were injected with platypus venom under the skin, they showed relatively mild effects. But when the same venom was injected directly into a vein, the animals died. This suggests that the venom could potentially be lethal to smaller animals, particularly if a spur strike happens to hit a major blood vessel, which might explain why the venom evolved despite platypuses not being hunters of large prey. The evolutionary history of venom in monotremes is fascinating. Many ancient mammal groups appear to have possessed venomous spurs, suggesting that this is not a feature unique to platypuses, but rather an ancestral trait that most mammals lost over time. The platypus, then, is not an anomaly that evolved venom, but rather a living fossil that retained it. This places the platypus's venom system in a completely different context. It's not a bizarre innovation, but a window into what the earliest mammals might have been like. Researchers are now studying platypus venom for medical applications. The same properties that make it so painful, particularly its resistance to standard painkillers, might teach us something about pain pathways in general, and potentially lead to better pain management strategies. Additionally, one of the proteins in platypus venom has shown promise in diabetes research, as it appears to have properties that could help regulate blood sugar. The platypus, in defending itself and competing with rivals, may inadvertently provide us with tools to fight some of humanity's most challenging medical conditions. But we should remember that this remarkable chemical arsenal is becoming increasingly rare. As platypus populations decline, each population lost represents not just the loss of individual animals, but the loss of millions of years of evolutionary refinement, a unique chemistry that exists nowhere else in nature, and that we've only barely begun to understand. Perhaps no feature of the platypus has captured the imagination quite like its method of reproduction. For here we encounter something that seems to violate one of the basic definitions of what it means to be a mammal. Mammals, we're taught, are warm-blooded, fur-bearing animals that give birth to live young and nurse them with milk. The platypus has fur. It's warm-blooded, more or less. It nurses its young with milk. But it lays eggs. For decades after the first platypus specimens reached Europe, this fact was hotly disputed. Surely, naturalists argued, this animal must give birth to live young like other mammals. The presence of mammary glands proved it was a mammal, and mammals didn't lay eggs. Even the suggestion that they might was met with ridicule from some corners of the scientific establishment. The debate was finally settled in 1884, when a young scientist named William Caldwell, working in Australia, sent a now famous telegram to the British Association. Monotremes oviparous, ovum meroblastic. In simple terms, monotremes lay eggs, and those eggs divide like reptile eggs. The news caused a sensation. The impossible was confirmed as fact. The reproductive cycle of the platypus is tied closely to the seasons, though the exact timing varies across the species range. In the warmer northern parts of Australia, mating occurs as early as late winter. In the cooler southern regions and Tasmania, it happens in spring, from August through October. This variation ensures that the young emerge from their mother's burrows when conditions are most favourable for their survival. The process begins with courtship, though relatively little is known about this phase of platypus life, since the animals are so secretive and most activity occurs in the water at night. What observations have been made describe it as a rather vigorous affair. A male platypus will pursue a female, grasping her tail in his bill while she leads him on an extended chase through the water. They may swim in circles together, roll over each other, and engage in various forms of aquatic acrobatics. These courtship rituals can last anywhere from a few minutes to over half an hour, and may be repeated over several days before mating actually occurs. The male takes no part in raising the young. Indeed, shortly after mating, the male and female part ways, likely never to interact again. The female is left to face the enormous task of reproduction and child-rearing entirely on her own, a pattern common among solitary mammals. After mating, the fertilized eggs develop inside the female for approximately 16 days, a period known as gestation. This is significantly shorter than the gestation period of comparable placental mammals. But that's because much of the development will occur outside the mother's body, inside the egg. During this time, the female begins the monumental task of excavating a nesting burrow. These burrows are different from the simple resting burrows that platypuses use year-round. A nesting burrow can be 30 meters long, winding deep into the riverbank, with multiple chambers and a special nesting chamber at the end. The female digs this entirely by herself, using her strong front claws to excavate soil and her flat tail to push the debris out behind her. she lines the nesting chamber with leaves, grass, and other vegetation, creating a soft, insulated space for her eggs. Once the burrow is complete, the female enters and seals herself inside by building a plug of soil in the tunnel behind her. This serves multiple purposes. It prevents predators from easily accessing the eggs and young. It helps maintain stable temperature and humidity in the nesting chamber. And it may also help keep the mother focused on the task ahead, preventing her from abandoning the nest during the long weeks of incubation and nursing. The female typically lays 1 to 3 eggs, though 2 is most common. The eggs are small, measuring roughly 17 millimeters in diameter, about the size of a marble. They have a soft, leathery shell, white in color, with a papery or parchment-like texture, quite unlike the hard-shelled eggs of birds, but similar to those of many reptiles. When the eggs are laid, they're slightly sticky, which helps them adhere to each other and to the mother's fur. The female then curls her body around the eggs, holding them between her abdomen and her tail, keeping them warm with her body heat. This external incubation lasts approximately 10 days, during which the mother rarely leaves the nest. She survives on the fat reserves stored in her tail and other parts of her body, reserves she built up before entering the nesting burrow. Inside the eggs, the developing platypus embryos grow rapidly. When they're ready to hatch, they use a specialized egg tooth, a small horny projection on the snout, to break through the leathery shell. They also have a fleshy nub called a carbuncle that helps in the hatching process. Both of these structures are evolutionary holdovers from reptilian ancestors and fall off shortly after hatching. The newly hatched platypus, called a puggle, is tiny, helpless, and profoundly altricial, meaning it's born in a very undeveloped state. It measures roughly 2 centimeters in length, about the size of a lima bean. It's blind, hairless, and completely dependent on its mother. In fact, the puggle is so underdeveloped at hatching that it would be considered almost embryonic by the standards of placental mammals. If the platypus gave live birth at an equivalent stage of development, it would essentially be birthing a fetus. Despite being so underdeveloped, the puggle has strong front limbs and can already crawl. It needs to, because it must make its way from the eggshell to its mother's belly to begin nursing. And this is where we encounter another of the platypus's remarkable peculiarities. The platypus has no nipples. Instead, the mammary glands open directly onto the skin through enlarged pores, located in 2 distinct mammary patches on the mother's abdomen. When the young nurse, they essentially lick milk that oozes through the mother's fur. The milk itself is rich and nutritious, containing all the proteins, fats, and other compounds necessary to support rapid growth, including antimicrobial properties that help keep the young healthy in the confined, warm environment of the nesting burrow. The young remain in the burrow with their mother for 3 to 4 months, growing rapidly. Their weight can increase 20-fold in the first 14 weeks of life. During this time, they develop fur, their eyes open, and they gradually become more coordinated and active. The mother periodically leaves the burrow to forage, building up the energy reserves she needs to continue producing milk. When she leaves, she may plug the burrow entrance again to protect her young. Eventually, around the time they're about 85% of their adult size, the young platypuses emerge from the burrow for the first time. They're fully furred, well coordinated, and ready to enter the water. Remarkably, they appear to learn to swim and hunt entirely on their own, through trial and error, without any teaching from their mother. Shortly after emerging, the young become independent, dispersing to find their own territories. Both males and females become sexually mature at around 2 years of age, though many females don't breed for the first time until they're 4 years old, or even older. Once mature, a female may breed annually, though studies have found that in any given year, fewer than half of adult females in a population actually reproduce. The decision to breed appears to depend on conditions in the months before mating season. If food has been plentiful and a female has been able to build up sufficient fat reserves, she's more likely to attempt breeding. If conditions have been poor, she may skip that year entirely, a strategy that ensures she doesn't commit to reproduction when success is unlikely. The platypus's egg-laying reproduction, combined with milk production, places it at a unique position in mammalian evolution. The genes responsible for producing milk are highly conserved across all mammals, suggesting that milk production evolved before the common ancestor of all living mammals. But the genes involved in live birth and placental development are absent in platypuses, confirming that these features evolved later, after the monotreme lineage had already branched off from the rest of the mammalian family tree. Interestingly, even though adult platypuses are toothless, they retain the genes for producing tooth enamel. Young platypuses are actually born with vestigial teeth, small non-functional teeth that serve no purpose and are soon lost. The fact that these genes still exist suggests that the platypus's toothless condition is a relatively recent evolutionary development, geologically speaking, and that their ancestors had functional teeth, a fact confirmed by fossil evidence. To truly understand the platypus, we must travel back in time, far beyond the reach of recorded history, beyond even the span of our own species' existence. We must go back more than 100 million years, to an age when dinosaurs still ruled the Earth, and the continents were arranged in configurations almost unrecognizable to modern eyes. During the Cretaceous period, roughly 110 to 146 million years ago, the first platypus-like animals appeared in the fossil record. These early monotremes lived in a world very different from our own. Australia, Antarctica, and South America were all joined together in a vast supercontinent called Gondwana. The climate was warmer, the sea levels higher, and the cast of characters populating the landscape was utterly alien to what we see today. The earliest known monotreme fossil is a tiny creature called Teinolophos trusleri, about the size of a modern rat. Its fossilized remains date from approximately 115 million years ago. This animal appears to have been terrestrial rather than aquatic, lacking the specialized bill and other aquatic adaptations we see in modern platypuses. It had teeth, small but functional, suggesting it fed on insects and other small prey. Another ancient monotreme from this era is Steropodon galmani, known from a beautifully preserved opalized jaw found in Australia. This creature was larger than Teinolophos and shows some features that suggest it might have been semi-aquatic. Though without a complete skeleton, it's hard to be certain. What's clear is that even this far back, the monotreme lineage had already diverged from the ancestors of all other mammals, following its own unique evolutionary path. The oldest animal that is definitely a member of the platypus family, based on teeth and leg bones, is Monotrematum sudamericanum. This species lived in Patagonia, in what is now southern Argentina, about 61 to 63 million years ago. At that time, South America and Australia were still connected through Antarctica, allowing animals to move between continents. Monotrematum was larger than modern platypuses, measuring roughly 70 centimeters in length. Its existence on a different continent tells us that platypus-like animals once had a much wider distribution than they do today. Other ancient platypus relatives have been found in Australia itself. Obdurodon, a genus known from several species spanning millions of years, had teeth and was larger than modern platypuses. The most impressive of these was Obdurodon tharalcuscheilde, which lived in the Miocene epoch around 15 million years ago. This giant platypus measured up to a meter in length and had powerful teeth capable of crushing hard-shelled prey. It likely hunted in open water rather than bottom-feeding in murky rivers like its modern descendants. The fossil record shows a clear trend in platypus evolution. The ancient forms were larger, had teeth, and likely had different ecological roles than modern platypuses. Over millions of years, the lineage became smaller, lost its teeth, and became increasingly specialized for bottom feeding in turbid freshwater environments. This specialization brought with it the refinement of electroreception and the development of the remarkable bill we see today. But the platypus's evolutionary story extends even further back. Molecular studies examining the DNA of living platypuses and comparing it to other mammals suggest that the monotreme lineage diverged from the ancestors of all other mammals, somewhere between 160 and 220 million years ago. This was during the Jurassic period, an age when the first mammals were tiny, shrew-like creatures living in the shadows of dinosaurs. This ancient divergence means that platypuses have been evolving independently, following their own path for an immense span of time. They've survived countless environmental changes, the rise and fall of dinosaurs, the breakup of Gondwana, multiple ice ages, and the dramatic climate shifts of recent geological history. They are, in a very real sense, living fossils, though that term should be used carefully. The platypus is not unchanged from ancient times, but rather is a highly specialized, modern animal that has retained certain primitive characteristics while also evolving unique adaptations all its own. One of the most fascinating aspects of platypus evolution revealed by modern genetics is the sex chromosome system. Most mammals, including humans, have a simple XY system for determining sex. Males have one X and one Y chromosome, while females have two X chromosomes. The platypus, however, has 10 sex chromosomes, 5 X chromosomes and 5 Y chromosomes in males, and 10 X chromosomes in females. During reproduction, these chromosomes form a chain during cell division, separating into two groups that segregate into sperm cells. Some sperm cells carry all 5 X chromosomes, while others carry all 5 Y chromosomes. When these combine with an egg that carries 5 X chromosomes, the resulting offspring is either male or female, depending on which type of sperm fertilized the egg. Even more remarkably, some of the platypus sex chromosomes show similarities to the sex chromosomes of birds rather than mammals. This suggests that the sex determination system we see in placental mammals evolved after monotremes had already branched off, and that monotremes retained a more ancient system that has parallels with what we see in modern birds. The platypus genome fully sequenced in 2008, has provided a treasure trove of insights into mammalian evolution. It contains a mixture of genes that are recognizably mammalian and genes that show reptilian characteristics. The genes for producing milk are there, highly conserved and similar to those in other mammals. But the genes involved in live birth, placental development, and other features we associate with typical mammals are either absent or non-functional. The genome also revealed fascinating details about the platypus's venom. The genes that produce venom proteins show signs of having been co-opted from the immune system, a process called recruitment. Many of the venom genes are related to defensin genes, which in other mammals produce antimicrobial proteins as part of the immune response. In the platypus, these genes have been duplicated, modified, and repurposed to create venom, an elegant example of evolutionary tinkering. Comparing the platypus genome to that of its closest living relative, the echidna, suggests that these two groups diverged from a common ancestor roughly 19 to 48 million years ago, though the exact timing is debated. The echidnas, with their spiny covering and terrestrial lifestyle, have taken a very different evolutionary path from the platypus, yet they share many fundamental features. They lay eggs, produce milk without nipples, have a cloaca, and possess electroreceptors, though far fewer than platypuses. The monotremes as a whole represent one of the three major branches of mammalian evolution. The other two are the marsupials, which include kangaroos, koalas, and opossums, and the placentals, which include everything from mice to whales to humans. Of these three branches, the monotremes are by far the oldest, having split off first. This makes them invaluable for understanding what the earliest mammals were like, and how the features we think of as defining mammals evolved over time. Sadly, the diversity of monotremes has declined dramatically. Once, there were many different species of platypus-like animals, inhabiting various ecological niches across the southern continents. Now, only 5 species remain: the platypus and 4 species of echidna. The platypus is the sole survivor of its family, a lonely branch on the great tree of life, the last echo of an ancient radiation that once filled niches we can now only imagine. As the sun sets over an eastern Australian river, the world transforms. The heat of day gives way to cool evening air. The surface of the water shifts from bright to dim, then dark. Most diurnal animals retreat to shelter, while the creatures of the night emerge. In a burrow carved into the riverbank, tucked beneath overhanging roots, a platypus stirs. This burrow is a simple affair, unlike the elaborate nesting burrows used for raising young. It's a short tunnel, perhaps 2 to 3 meters long, ending in a chamber just large enough for the platypus to curl up and sleep. The entrance is typically below the waterline, hidden from casual observation, accessible only by diving. The platypus emerges, sliding smoothly from the burrow into the water. This is a male, fully grown, weighing about 5 pounds. His coat is thick and dark, perfectly waterproof. As he swims, his eyes are open, taking in what little light remains. His ears and nostrils are also open, gathering information about the world above the surface. But soon, he will close all of these and navigate by senses we cannot share. He swims to the surface, takes a breath, and dives. The dive is smooth, practiced, his back arching characteristically as he plunges down. His front feet paddle in alternating strokes, webbing fully extended. His hind feet and tail serve as rudders, guiding him toward the river bottom. Within seconds, he's there, 5 feet down, in water that's cool and clouded with sediment from recent rains. Now, everything changes. His eyes close, his ears seal shut, his nostrils clamp tight. He's holding his breath, and he'll remain submerged for about 40 seconds, though he's capable of staying down for longer if needed. In this sensory isolation, most animals would be helpless, but the platypus is in his element. His bill sweeps from side to side, scanning the muddy bottom. The tens of thousands of receptors in the bill are active, painting a picture of the underwater world in electrical and mechanical patterns. There, a few inches to the left, a freshwater shrimp has just flicked its tail, trying to escape. The platypus detects both the electrical pulse from the muscle contraction and the physical disturbance in the water. By comparing these signals, he knows exactly where the shrimp is and how far away. He adjusts his trajectory, moving smoothly through the water. His bill probes into the mud, grasps the shrimp in the soft, rubbery edges, and brings it up into his cheek pouches. The shrimp is still alive, still struggling, but it's caught. The platypus continues his dive, scanning, probing, finding more prey. A caddisfly larva hiding in a case of tiny pebbles, a small worm, a water beetle. After about 40 seconds, his oxygen reserves are depleting. He returns to the surface, arching upward with powerful strokes. He breaks through, breathing, the air sweet and necessary. While floating at the surface, he begins to grind his catch between the horny pads in his bill, macerating the food mixed with bits of gravel he scooped up from the bottom. The gravel helps grind the food into a paste he can swallow. Then he dives again. This pattern continues for hours. Dive, forage, surface, grind, breathe, dive again. A platypus may spend 10 to 12 hours foraging each night, making hundreds of dives. During this time, he can consume prey equal to about half his body weight, a remarkable amount that testifies to the energy demands of his lifestyle. The food he's collecting is varied. Insect larvae make up the bulk of his diet, particularly mayfly, caddisfly, and beetle larvae. But he'll also eat freshwater crustaceans like shrimp and crayfish, aquatic worms, small mollusks, and occasionally fish eggs or small fish. The exact composition of his diet varies with season and location, depending on what's available in his particular stretch of river. As the night progresses, he moves up and down his territory, a section of river, perhaps a kilometer or two in length. This is his home range, an area he knows intimately, every pool and riffle, every snag and rock. His territory overlaps with those of several females, which is intentional. Males establish territories that encompass the ranges of multiple females, ensuring access to potential mates when breeding season arrives. But he's not alone in these waters. Other predators hunt here too. A water rat passes by, swimming near the surface. The platypus pays it little attention. They're not competitors, as the rat hunts different prey. More concerning are the larger fish. Murray cod, massive predatory fish that can exceed a meter in length, are known to occasionally take platypuses. Birds of prey, like white-bellied sea eagles, hunt during the day and have been known to catch platypuses that venture out too early or too late. On land, foxes and dingoes pose a threat, particularly to young platypuses making their first uncertain forays from the nesting burrow. As dawn approaches, the platypus's foraging becomes less intense. He's eaten well tonight, his cheek pouches repeatedly filled and emptied, his stomach now comfortably full. The sky begins to lighten. While platypuses are primarily nocturnal, they can be active during the day, particularly during the winter months or on overcast days. But this platypus is ready to rest. He returns to his burrow, diving down to the entrance, swimming into the tunnel. Inside, safe from predators, he grooms his fur carefully. This grooming is essential. His fur must remain waterproof, with the long guard hairs protecting the soft underfur beneath, and the air layer trapped between skin and fur maintained. He uses his bill and his front claws to work through his coat, redistributing oils, removing debris, ensuring everything is in order. Finally, he curls up in the sleeping chamber, his tail touching his bill, forming a tight ball that conserves heat. His metabolic rate drops slightly. His body temperature, already lower than most mammals, falls a bit further. He sleeps. Outside, the river continues its eternal flow. The sun rises, the day creatures take over from the night creatures, and the great cycles of nature turn. The platypus sleeps through it all, resting, digesting, preparing for another night of hunting in the dark, cold waters of his ancient home. This is the pattern of his life, night after night, through the changing seasons. In winter, when the water is coldest, his dives may be shorter, his foraging more intense, the energy demands of maintaining body temperature higher. In summer, when the water warms and prey is more abundant, life is somewhat easier. Through it all, he persists, following patterns laid down by millions of years of evolution, a living link to the deep past, carrying on in a modern world that grows ever more challenging for his kind. In 2020, scientists made a discovery about the platypus that seemed almost too strange to believe. Given everything else we know about this animal, that's saying something. The discovery happened, as many great discoveries do, largely by accident. A team of researchers at Northland College in Wisconsin was studying biofluorescence in flying squirrels, documenting how these animals glow pink under ultraviolet light. While examining preserved squirrel specimens in the Field Museum in Chicago, they wondered what other mammals might share this trait. Almost on a whim, they turned their UV lights on a preserved platypus specimen in a nearby drawer. The platypus glowed. Under normal visible light, the platypus appeared exactly as expected. A uniform brown, perhaps a bit duller than a living specimen, but otherwise unremarkable. But when illuminated with ultraviolet light at wavelengths of 385 to 395 nanometers, the fur emitted a vivid blue-green glow, with peak emissions around 500 nanometers. The researchers could hardly believe what they were seeing. They examined 3 different specimens, males and females, from different locations in Australia. All of them showed the same effect. The fur absorbed ultraviolet light and re-emitted it as visible light in the blue-green range, a phenomenon known as biofluorescence. The pattern was consistent across the body, suggesting this wasn't just contamination or an artifact of preservation. This was a real property of platypus fur. Biofluorescence is not the same as bioluminescence. Bioluminescent animals, like fireflies or certain deep-sea fish, produce their own light through chemical reactions. Biofluorescent animals absorb light at one wavelength and re-emit it at a different wavelength. They don't generate light, they transform it. Before this discovery, biofluorescence was known in only a handful of mammals. Flying squirrels, which glow pink under UV light, had been documented just a year earlier. Some species of opossum also show this trait, glowing in various shades of pink and purple. But these were placental and marsupial mammals. The platypus, as a monotreme, represented a completely different branch of the mammalian family tree, one that had diverged more than 150 million years ago. The fact that such distantly related mammals all showed biofluorescence suggested several possibilities. It could be an ancient trait present in the common ancestor of all mammals and retained in these few modern species. Or it could be a trait that evolved independently multiple times, a case of convergent evolution. Or it could simply be a byproduct of fur chemistry with no adaptive significance at all. But why would a platypus glow? That's the question that has no definitive answer yet, only hypotheses. The researchers who made the discovery suggested several possibilities. One idea relates to predator avoidance. Many of the platypus's predators, including large predatory fish and various birds, can see in the ultraviolet range. Most birds, in fact, have 4 types of color receptors compared to the 3 that humans have, with the 4th type tuned to UV wavelengths. For these animals, the ultraviolet part of the spectrum is as visible as blue or red is to us. By absorbing UV light and re-emitting it as blue-green, the platypus might be altering its visual signature in ways that make it harder for predators to detect or identify, particularly in the dappled moonlight and dawn or dusk lighting conditions when platypuses are most active. Another hypothesis suggests that biofluorescence might play a role in intraspecific communication, helping platypuses recognize each other in low light conditions. Platypuses are primarily solitary, but they do interact during breeding season. If they can see ultraviolet light, which is still uncertain, the fluorescent glow might serve as a species-specific signal, a way of identifying another platypus in the murky waters or shadowy banks of their riverine habitat. A third, more prosaic explanation is that the biofluorescence is simply an incidental byproduct of the chemistry of the fur. Certain compounds naturally present in the fur might absorb and re-emit light without this having any functional significance. The trait could be neutral, neither helpful nor harmful, simply existing because there's no strong selection against it. To understand which of these explanations, if any, is correct, we need to know whether living platypuses show the same biofluorescence as preserved specimens. Museum specimens are treated with various chemicals and may degrade in ways that could alter their fluorescent properties. Shortly after the initial discovery was published, another group of researchers in Australia examined a freshly killed platypus, a road-killed specimen that had not been preserved or chemically treated. It too glowed under UV light, confirming that this is a real property of living platypuses, not an artifact of museum preservation. We also need to know whether platypuses can see ultraviolet light. If they can't, then the communication hypothesis becomes less likely, though the predator avoidance idea could still hold. Studies of platypus eyes suggest they have relatively simple vision, adapted more for detecting movement and general shapes in low light than for color discrimination or detailed imagery. Whether they possess UV-sensitive photoreceptors is still being investigated. The molecular basis of the biofluorescence is another open question. What compounds in the fur are responsible for absorbing and re-emitting light? Preliminary studies suggest that tryptophan, an amino acid, and certain keratin structures might be involved, but the full picture is not yet clear. Understanding the chemistry could help determine whether this trait is likely to have functional significance, or is simply an incidental property of the materials that make up platypus fur. What we can say with certainty is that this discovery adds yet another layer to the already remarkable story of the platypus. An animal that lays eggs, produces venom, detects electricity, and navigates by senses alien to us, also glows in the dark when illuminated by ultraviolet light. It's as if nature, having already created something unprecedented, decided to add one more impossible flourish. And it raises tantalizing questions. How many other animals might have biofluorescent properties we haven't discovered yet? If we were to go through museum collections around the world, shining UV lights on preserved specimens, what other surprises might we find? The natural world, it seems, still holds secrets we've literally overlooked, hidden in plain sight, visible only when we learn to look in new ways. For the platypus, this discovery underscores how much we still don't understand about even well-studied species. Despite centuries of scientific investigation, despite extensive research into its anatomy, physiology, behavior, and ecology, the platypus can still surprise us. It reminds us to remain humble in the face of nature's complexity, to keep asking questions, and to never assume we've learned all there is to know. For millions of years, the platypus has survived. It weathered the extinction of dinosaurs, the breakup of continents, multiple ice ages, and profound climate shifts. It adapted to changes that wiped out countless other species. But now, in the geological blink of an eye, it faces challenges unlike any it has encountered before. The platypus is currently listed as near threatened by the International Union for Conservation of Nature, a classification that suggests the species is not yet endangered, but is heading in that direction. In some parts of its range, the situation is more dire. In South Australia, where platypuses were once common along the Murray River, and in the Adelaide Hills, the species is listed as endangered. Only a handful of individuals remain in isolated pockets. In Victoria, the species was recently listed as vulnerable after evidence mounted that populations were declining significantly. The numbers tell a sobering story. Over the past 30 years, platypus habitat in Australia has shrunk by at least 22%. That's an area roughly 3 times the size of Tasmania, lost to this species in just 3 decades. In some river systems, platypuses have disappeared entirely. In others, populations have crashed to a fraction of their former abundance. A comprehensive study, published in 2020, examined the risk of extinction facing platypuses. The researchers looked at current threats and projected how platypus populations would fare over the next 50 years under various scenarios. Their findings were alarming. Under current conditions, considering water extraction, land clearing, climate change, and other ongoing threats, platypus abundance was predicted to drop by 47 to 66%. Local extinctions would occur across approximately 40% of the species' range. If climate projections for 2070 are included, accounting for increased drought frequency and severity, the situation becomes even more dire. Platypus abundance could decline by 51 to 73%, with metapopulation occupancy, a measure of how many suitable habitat patches remain occupied, dropping by 36 to 56%. These numbers suggested that the platypus could soon qualify for listing as vulnerable under the criteria used by the IUCN. What's driving these declines? The threats are multiple and interconnected, a web of human impacts that together are making platypus habitat increasingly inhospitable. water extraction is one of the most significant threats. Platypuses require permanent fresh water, year-round flow in the rivers and streams they inhabit. But throughout eastern Australia, water is being extracted for agriculture, urban use, and industry at unsustainable rates. Dams regulate river flows, often reducing them dramatically during dry periods. Irrigation channels divert water away from natural waterways. The result is that many rivers that once flowed perennially now dry up seasonally, leaving platypuses stranded without the water they need to survive. Habitat degradation goes hand in hand with water extraction. The riparian zones, the vegetated areas along riverbanks, are critical for platypuses. These zones provide shade, stabilize banks, prevent erosion, contribute organic matter to the water, and support the aquatic invertebrates that platypuses eat. When riparian vegetation is cleared for agriculture or development, when banks are stabilized with concrete or rock rather than living plants, when livestock are allowed to graze right to the water's edge, the habitat becomes unsuitable for platypuses. Climate change amplifies all of these problems. As temperatures rise and rainfall patterns shift, droughts become more frequent and severe. The catastrophic bushfires of 2019 and 2020 devastated large areas of eastern Australia, including significant platypus habitat. In the 9 months following those fires, platypus populations in affected areas declined by 14 to 18%. The fires themselves killed some animals directly, but the longer-term impacts were even more severe. Ash and sediment washed into waterways, smothering aquatic invertebrates and reducing food availability. Vegetation that once shaded streams and stabilized banks was destroyed, leaving the habitat exposed and degraded. Pollution poses another serious threat. Agricultural runoff carries pesticides, herbicides, and excess nutrients into waterways. Urban stormwater brings oils, heavy metals, and other contaminants. These pollutants can directly harm platypuses, affecting their health and reproductive success. They can also devastate the aquatic invertebrate populations that platypuses depend on for food. Studies have found that platypuses are largely absent from urban and agricultural streams with high levels of pollution, even when other habitat features seem suitable. Fishing gear and yabby traps, particularly a type called opera house traps, can drown platypuses. These dome-shaped traps are used to catch freshwater crayfish, but have no escape mechanism for air-breathing animals that become trapped inside. Platypuses enter the traps while foraging and are unable to reach the surface to breathe. Several Australian states have banned these traps or restricted their use, but drowning in fishing gear remains a documented cause of platypus mortality. Introduced predators, particularly foxes, pose a significant threat, especially to young platypuses. Foxes hunt along riverbanks and can catch platypuses when they come ashore. Native predators like quolls and goannas also take platypuses opportunistically. But these native predators have always been part of the ecosystem. Foxes, introduced by European settlers, represent a new and substantial threat. The loss of connectivity between populations is perhaps one of the most insidious threats. As platypus populations become fragmented, separated by dams, dry river sections, or degraded habitat, they become isolated. Isolated populations are more vulnerable to local extinction from random events. They're also more prone to inbreeding depression, where the lack of genetic diversity leads to reduced fitness and health. Platypuses are not good dispersers overland. They travel along waterways, When a river is broken up by dams or dry sections, platypuses in one segment can't easily reach another segment to recolonize after a local extinction. Given all these threats, what's being done to protect platypuses? The answers are encouraging in some ways, insufficient in others. Legally, platypuses are protected throughout their range. It's illegal to harm, capture, or kill them without special permits. However, legal protection doesn't prevent habitat destruction or address most of the threats facing the species. Captive breeding has been attempted, with mixed success. Several facilities in Australia, including Healesville Sanctuary in Victoria, have successfully bred platypuses in captivity. This is extraordinarily difficult due to the platypus's specialized requirements and sensitivity to stress. But when it succeeds, captive breeding provides a potential insurance population and can produce animals for reintroduction programs. Reintroduction efforts are underway in some areas. In 2023, a major project began to reintroduce platypuses to Royal National Park, near Sydney, an area where they had gone locally extinct. Similar efforts are being considered for other parts of the range where platypuses have disappeared, but suitable habitat could potentially be restored. Habitat restoration is crucial. This includes revegetating riparian zones, removing or modifying dams to restore more natural flow patterns, reducing pollution, and protecting remaining high-quality habitat from development. Several organisations, including the Australian Platypus Conservancy, WWF Australia, and various government agencies, are working on habitat restoration projects throughout the platypus range. Research continues to be essential. We still don't know basic facts about platypus populations, abundance, and distribution in many areas. Citizen science programs, where members of the public report platypus sightings, help fill these knowledge gaps. Environmental DNA techniques, where water samples are analysed for trace platypus DNA, provide a non-invasive way to detect platypus presence. Long-term monitoring programs track population trends and help identify threats before they become crises. Public awareness is growing. The platypus is an iconic species, beloved both in Australia and internationally. When people learn that this remarkable animal is in trouble, many want to help. Conservation organisations are working to channel this public concern into action, whether through donations, volunteer work, or advocacy for stronger environmental protections. But the fundamental challenge remains. Saving the platypus means addressing the broader environmental issues facing Eastern Australia's freshwater ecosystems. It means rethinking water management to balance human needs with ecological requirements. It means controlling land clearing and agricultural runoff. It means addressing climate change, the overarching threat that amplifies all other problems. The platypus survived the extinction of the dinosaurs. It persisted through ice ages and the fragmentation of Gondwana, but it may not survive us. The threats it faces now are different from anything in its long evolutionary history. They're rapid, pervasive, and driven by a single species that has, in just a few centuries, reshaped the planet to a degree unprecedented in Earth's history. Whether the platypus has a future depends on the choices we make now. Every dam we build or remove, every river we restore or degrade, every patch of riparian forest we protect or clear, matters for this species. The platypus can't adapt to our changes fast enough. Evolution works over millions of years. We're transforming the world in decades. As we come to the end of our journey through the world of the platypus, it's worth reflecting on what this animal represents. In a purely scientific sense, the platypus is important because of what it can teach us about evolution, about how mammals diversified, about the origins of features we take for granted, like live birth and lactation. Its genome has provided invaluable insights. Its venom offers potential medical applications. Its electroreception demonstrates possibilities we might never have imagined. But the platypus is more than a scientific curiosity, more than a collection of useful genes or interesting adaptations. It's a reminder that the natural world is richer, stranger, and more wonderful than we often remember. In an age when we can sequence genomes and track animals with satellites, when we've mapped the ocean floor and photographed distant galaxies, it's humbling to realize how much we still don't understand about a mammal that lives in the rivers of eastern Australia. The platypus defies our categories. It doesn't fit neatly into the boxes we create to organize and understand the living world. It has a bill, but isn't a bird. It lays eggs, but is still a mammal. It has venom, but uses it for competition rather than hunting. It glows under UV light for reasons we can't fully explain. It detects electricity with senses we don't possess. It represents a successful evolutionary experiment that has persisted for over 100 million years, following a path utterly different from the one that led to us. In Aboriginal Australian mythology, there are various stories explaining the platypus's unusual appearance. One Dreamtime story tells of a young female duck who was abducted by a water rat. When she eventually returned to her tribe, she laid eggs, and from these hatched creatures that had their father's body and tail, and their mother's bill and webbed feet. The other animals at first refused to accept these strange offspring, but eventually welcomed them, and they became the platypuses. This story, like the scientific account, recognizes that the platypus is different, that it combines features in unexpected ways. But the story also contains an important message about acceptance, about finding a place for things that don't fit our preconceptions. In the Dreamtime story, the other animals initially reject the platypuses because they're strange, but they come to accept them, to welcome them as part of the community of living things. We would do well to remember this lesson. The platypus has a right to exist, not because it's useful to us, not because it might provide medical breakthroughs or tourist revenue, but simply because it is. It's a unique expression of life's diversity, a result of millions of years of evolution, adapted to its ecological niche in ways both beautiful and bizarre. When we protect the platypus, we're not just saving one species. We're protecting the rivers and streams it inhabits, the complex web of invertebrates and fish and plants that make up its ecosystem. We're preserving genetic diversity that took eons to develop. We're maintaining ecological processes that we may not fully understand, but that are nonetheless crucial to the functioning of these freshwater systems. And we're keeping wonder alive in the world. In a time when so much seems known, quantified, and explained, the platypus reminds us that mystery endures. There are still discoveries to be made, questions to be answered, secrets waiting to be uncovered in the bend of a river or the depths of a muddy bottom. The platypus is ancient and modern, primitive and specialized, familiar and alien. It's a reminder that evolution doesn't move in a straight line toward complexity or perfection, but branches in countless directions, finding solutions to the challenges of survival in ways we might never expect. As you drift off to sleep tonight, Perhaps you'll dream of dark rivers in Australia, of an animal with a bill like a duck and fur like an otter, swimming through waters we can barely imagine, navigating by electricity, carrying in its body the echoes of an ancient past and the hopes for an uncertain future. The platypus persists, for now, in its rivers and streams. May it persist for millions of years more, continuing to surprise us, to challenge our understanding, to remind us that the living world is more remarkable than we can ever fully comprehend. If you enjoyed this exploration of one of nature's most remarkable creatures, please consider liking this video and subscribing to Bedtime Biology. Share your thoughts in the comments below. What surprised you most about the platypus? What other creatures would you like us to explore? Your support helps us continue bringing you these calm, curious journeys through the wonders of the natural world, one sleepy discovery at a time. Until next time, sleep well, dream deeply, and stay curious about the incredible life that shares our planet.